Welcome to part 2 of the lecture series dealing with the immune. So what exactly do I mean by alternation in life cycle stages? Well, let's go ahead and look at Plasmodium, which causes the disease known as malaria. Again, malaria is the #1 cause of parasite-induced death worldwide, and so it does in fact produce or provide a very serious problem overall worldwide. What you see in this particular slide is the life cycle stages associated with Plasmodium. So if you look at the very top, what you can see is that the pathogen lives inside of the mosquito gut, and that's actually on the left-hand side of the slide. This pathogen lives inside the mosquito gut. The mosquito is a vector that carries this particular pathogen to the host, in this case a human. When the mosquito goes for a feed, the blood is drawn from the host individuals, and in that process, what happens is the pathogen then enters into the host system. At this particular point, the life cycle stage is known as a sporozoite and has specific antigens associated with that life cycle stage. Those sporozoites travel to the liver, where they convert into a different life cycle stage known as merozoites. The merozoites leave the liver, enter into the bloodstream, and infect red blood cells. In the red blood cells, we know that we have another change in the life cycle to produce the gametocytes. Once the gametocytes are produced, then another mosquito can do a blood feed and transmit those gametocytes into the gut of the mosquito, and hence the life cycle continues for this particular parasite. Now, the problem is, again, or the way that these parasites typically evade the immune response is due to antigenic variation. What I've listed here are 3 different life cycle stages for malaria, and with those different life cycle stages, there are different antigens that are produced. So, if the host immune system first recognizes the sporozoite stage and an immune response is elicited against the sporozoites, for example, the adaptive immune response with antibody production, what happens is those antibodies are produced and are used to then target the sporozoites. However, once the sporozoites enter the liver, they change their life cycle stage, hence they change their antigens. Those antibodies that were produced against the sporozoites no longer detect or interact with the antigens that are now present on the merozoites. And so this is a continual problem overall, that the immune system is trying to play catch up with production of antibodies to antigens that are no longer present. This particular slide demonstrates or depicts that antigenic variation in a little different way. This happens to be for African sleeping sickness, but the concept is still the same. In the top portion of the slide, you see the parasite in either blue color, pink color, or green color, and these represent antigens that are present during different stages of the life cycle of the parasite. If you look down at the bottom, you can see a graph that shows the approximate time after the fly bite, whence the parasite is transmitted to the host. And you can see, as far as time is concerned, the variations that occur in the antigens depending upon the day of infection in the host. Now, again, it is this change then that causes a problem, because the antibodies that are produced against the first form, the blue form, for example, will no longer be able to interact with what was the blue form, because there has been a change or a shift in the life cycle stage to where that antigen is no longer present. Even though the antibodies have been produced, that particular antigen is no longer present. At that particular point, there is a different antigen that is present in the pink form, and our immune response is kicked into high gear. There's now antigens recognized for the pink form, and antibodies are produced. Those antibodies get produced and are used to fight off this particular pathogen. However, you can see then a few days later, there's a change again in the antigens that are present, Hence, the pink antigen is no longer present, so the pink antibodies have nothing to bind to, and hence this is the survival and the success of this particular pathogen. Single-celled parasitic pathogens are interesting in the fact that, again, there is differences in the location and where you can find the parasite itself. There are some that have extracellular stages, and you would imagine then with an extracellular type of parasite, single-celled parasite, that there's a strong humoral response that is elicited. That is, antibodies are playing a really big role in fighting off those types of parasites that have an extracellular stage. As you can see from this particular slide, there are also some parasites that have an intracellular stage, meaning there are some parasites that actually get inside of the host cells. Like we just discussed with the one particular life cycle stage for malaria, they actually enter into the red blood cell. So when you have a parasite that enters into the host cell, again intracellular, in this particular case, the strong response that is elicited is a cell-mediated response. Those cytotoxic T cells are elicited to help destroy those altered self cells. We've talked about altered self cells multiple times throughout the semester, and usually we refer to those altered self cells as cells that have been infected with the virus or those that are a tumor. But in this particular case, because there are some parasites that can be intracellular, they can also be host cells that are infected with a parasite. So now I want to focus on a few more specific evasion mechanisms. Plasmodium falciparum, which is the parasite that causes the disease malaria, its number one way of evading the host immune response is antigenic variation, and we looked at that on the previous slide. Trypanosoma brucei, which causes the African sleeping sickness, also has a strong antigenic variation mechanism for evading the host immune system. However, there are some other examples with this particular species of evasion of host immune response. This particular parasite can produce or is known to produce molecules that are very much like cytokines or act like cytokines. These cytokines, however, alter T and B cell propagation and proliferation. Hence, those B and T cell responses are diminished because of these molecules that are cytokine-like playing a role in the host immune system. Trypanosoma cruzi, which causes Chagas disease, also exhibits antigenic variation. As I mentioned at the beginning of the lecture, most single-celled parasites do in fact demonstrate this antigenic variation as a very good and strong mechanism to evade the host immune system. However, this particular parasite can also suppress T cells and play a role in binding to macrophages that then alter cytokine secretion. And lastly, Entamoeba histolytica is known to produce proteases. Proteases are large classes of enzymes that degrade proteins. More specifically, these proteases are known to degrade antibodies that may be produced or as an immune response against this particular pathogen. We also know that there are proteases that are produced by this pathogen that can inactivate complement system proteins. And we also know that there are proteases that can inhibit CAM adherence overall. And CAMs, again, are cell adhesion molecules that are going to play an important role in the extravasation process. So if we have a protease that then plays a role in destroying these CAMs, there's going to be a problem with getting those immune cells to the target location where an infection has taken place. Now, parasites can also be multicellular. In this particular slide, there are a number of different parasites listed here. All of them are multicellular except for the amoeba. The amoeba is in fact single-celled. Unfortunately, I couldn't find a nice figure that could show several multicellular parasites all at once. So just disregard amoeba because amoeba is in fact single-celled. So you see very clearly a division between the two categories of multicellular parasites. On the right, you have things like tick, louse, or flea. These are organisms that typically live on the exterior surface of the host system, they are still in fact considered parasites. So let's talk for a minute about the type of immune response that could be elicited against this particular type of parasite. If you think back to that response that we discussed, that the skin is acting as a physical barrier, it is in fact acting as a physical barrier, but all of those antimicrobial peptides that are being produced do in fact play a little bit of a role in fighting off these particular types of multicellular pathogens. Obviously, that response is not super strong because these are multicellular organisms, and typically those antimicrobial peptides are targeting microbial components, but there are in fact some enzymes that can play a role in trying to fight off somewhat these multicellular parasites that are on the exterior surface of the host. So on the left side of the slide, what you're looking at are multicellular parasites that are found inside the host system. And when we talk about multicellular parasites found inside the host, what we are mostly referring to are parasitic worms. Parasitic worms are categorized as helminths, and we can subdivide them into the roundworms, which are nematodes, or flatworms, which is the platyhelminthes. The flatworms can also be further subdivided into flukes and tapeworms. Flukes are going to be the ones that are found within the tissues of the host system, and tapeworms are going to be specifically found within the gut of the host system. Now, interestingly, with these particular multicellular parasites, again, we have them being eukaryotic, which makes a special problem for our immune system, like I mentioned previously. But in this case, these are multicellular organisms. They are large, and that is another extreme problem for our immune system to try and overcome. So with multicellular parasites, there is in fact a host immune response, but typically it is not sufficient to remove that particular pathogen from the host system or to remove the infection altogether. So when we talk about the evasion mechanisms that these multicellular parasites have that can be found inside the host system, like we just discussed, the flukes and the tapeworms, they are extremely successful because, again, an immune response is elicited, but it is not sufficient. With these multicellular parasites, most of them enclose themselves in a glycolipid glycoprotein coat that is derived from the host system, and this then provides some amount of protection from the immune response as well. So we have something that is relatively large in comparison to the cells that are trying to destroy it, the leukocytes, and these typically have an extra coating, glycolipid and glycoprotein coating, that then protect them as well. So let's go ahead and talk just a little bit about the immune response that is elicited against one of these large multicellular parasites. In this particular case, we're looking at an adult worm, a fluke, that is found in the tissue of the host. Where I have the number 1, what we see is that there is in fact an increase in the IgE class of antibodies. So if you recall, when we talked about antibody classes, IgE was specifically the class of antibody that is produced or elicited in response to a parasitic infection. And that is what we see happening at the top of the slide. For number 2, what we see is an increase in mast cells, and in particular the degranulation of mast cells releasing histamine and heparin. We know what histamine and heparin can do. We know that those are acting as inflammatory components or pro-inflammatory components that can help then allow for more cells to enter into the target location, which we see sort of down at the bottom of the slide. For number 3, we also see an increase at the tissue level of eosinophils. Again, going back to the cell types that we know are part of the immune response, eosinophils play a role, or we know are playing a role, in a parasitic response. In particular, they are playing a role in eliciting the ADCC mechanism, antibody-dependent cell cytotoxicity mechanism. So what you see at this particular point where the arrow is pointing is those antibodies binding to the antigen of the multicellular parasite, and the eosinophils coming in to help play a role in eliciting the ADCC response. We know, though, that cytokines are also playing a really big role in allowing for these immune cells to function in the way that they are functioning. For number 4, we see that infiltration of inflammatory cells. So again, cells that are elicited in this inflammatory response that we talked about for step 2. Those are the cells that are now entering that location where the infection is taking place. And then lastly, we also know there is in fact a helper T cell response, which again helps orchestrate all of these leukocytes to do exactly what they're trying to do, to play a role in targeting the destruction of this particular multicellular parasite. The last category or class of pathogen that I want to discuss are fungal pathogens. Fungal pathogens, as I mentioned at the beginning of the lecture, provide a unique problem for the host immune response. Unique in the sense that these cells are also eukaryotic, just like the host cells being eukaryotic. So there is overall less divergence between the two cell types, hence less immunogenicity in general. It doesn't mean that these are not immunogenic, but there are components that are going to be less immunogenic overall because the cell types are the same cell types overall. Also, I mentioned that fungal pathogens can be either single-celled or multicellular. This also presents a big problem for the host immune response, similar to what we discussed with parasitic infections in the previous slides. Fungal diseases are in fact very diverse because of the diverse types of fungi that we know exist. When we talk about fungal diseases, these are collectively referred to as mycoses and can be classified in 3 different broad ways. The disease or the mycosis can be classified because of the site of infection, where the infection takes place. So for example, if it's superficial, it could be on the epidermis, cutaneous, located within the skin, hair, or nails, subcutaneous, within a wound, for example, or deeper systemic, like within the lung or abdominal tissue. So again, the site of infection can be one way to classify mycoses. Mycoses can also be classified by the route of acquisition. How is the pathogen entering into the host system? Is it exogenous, something that is environmental, airborne, or cutaneous? Or is it something that is endogenous, something that's always present or commensal? Lastly, they can be categorized or classified based on their virulence. Something that's considered primary is going to be extremely virulent and infects healthy hosts, just about anyone, versus an opportunistic type of fungal pathogen typically has very low virulence and will typically infect immunocompromised hosts. As I mentioned on the previous slide, fungal pathogens are in fact very diverse, and you see some examples of fungal pathogens on this particular slide. Some can be single-celled, some can be multicellular, and we also know that fungal pathogens exhibit alternation in life cycle stages. I should say some fungal pathogens exhibit alternation in life cycle stages, so that key mechanism that we just discussed for parasites also holds true for certain fungal pathogens, and that there is antigenic variation that occurs with these particular pathogens, and that allows for a very strong evasion of the host immune response. So what type of host immune responses do we know come into play against fungal pathogens? Phagocytosis is definitely a key player in fighting off fungal pathogens, specifically single-celled fungal pathogens. We know that there are PRRs, in particular some of the toll-like receptors that have the ability to recognize PAMPs, and in this case, these PRRs do a really good job at controlling single-celled fungal pathogens. We also know that both the alternative and the MB-lectin-activated complement system can recognize cell wall components to target specific fungal pathogens. There are also plenty of examples of antibodies that have been detected against fungal pathogens, suggesting or indicating that there is, in fact, an adaptive immune response against fungal pathogens as well. So how are these fungal pathogens evading the host immune response? That whole mechanism of antigenic variation or antigenic variability plays a really big role in fungal invasion. We know that many fungi have the ability to produce a capsule, which will prevent PRR binding. Many fungi are known to adhere to surfaces within the host system. And so this congregation or this fungal adherence also plays a role in causing evasion of the host immune system. In addition, there are fungi that are known to produce phospholipases, which are broad categories of enzymes that degrade phospholipids, as well as proteases, like we discussed previously, that can then cause tissue damage and impair the host immune defense system. Lastly, I like to show this slide because it demonstrates two components on it. In the red coloring, you see newly emerging diseases, things like Ebola. On this particular slide, the SARS outbreak of 2002, and once this slide gets updated, probably our current SARS-CoV-2 outbreak. These are newly emerging diseases that our immune systems have not encountered and need to interact with to be able to elicit an immune response. We also have a problem worldwide with the diseases that are listed in black, and these are re-emerging. Re-emerging oftentimes due to vaccine lacks, things like whooping cough and measles, for example. The major anti-vax movement could be contributing overall to the increase or the reemergence of these particular diseases. What's causing both of these categories in general oftentimes is blamed on globalization, and interactions between people and wildlife are occurring at a much greater rate to potentially cause these problems overall. So are our immune systems perfect? No, they are not. And what we've hopefully learned from this series of lectures is that many of these pathogens have evolved ways to evade the host immune system, and so there's a constant battle between our immune response as well as the pathogens that we're trying to destroy.